Okay. Hi, everyone. Today we have Yicong Huang. Yicong is a PhD student in my lab. He's the first PhD student who joined my lab. He did his bachelor's in intelligence science and technology at Zhejiang University in China. And then he did 2 masters. One, a Master of Engineering at South China University of Technology in Guangzhou, China. And then he did another master's in computer science at Georgia Tech. Yicong joined my lab in 2023, and he's in the joint Georgia Tech and Emory Biomedical Engineering PhD program. His research interests are computational neuroscience and also neuroengineering, and he focuses on understanding the neural mechanisms that underlie learning and memory. This paper is very exciting to all of us because that's the first real paper from our lab with everything done in the lab. Okay, John, go ahead.
You can have my name.
Yeah.
Yeah. Hello everyone, I'm Yicong. Today I'm gonna present our first paper in the lab. And this paper was done not just me, but also many people in the lab. So for example, Ali is doing the initial analysis on the clustering, and Mengze and Shuang are the one there who runs the majority of the experience. So let me give you a little bit background first. So before we start, I want to introduce 2 concepts. One is the predictive processing. So this framework tells you that the brain can form an internal model of the world based on your prior experience. And from the internal model, it's gonna generate prediction to the sensory input, but not just passively receive it. So if there is a discrepancy between the actual sensory input and the predictions, there will be a prediction errors going backward to the internal model to update it. So this is the framework of how the brain is learning. The other concept is temporal processing, which means the brain can understand the timing of those temporal events. So it's not just the sensory input itself, but also the timing of the sensory input. So I'm giving you 2 example images here. So as you can see from the loop of the animation, you can kind of predict when you're gonna go for the next loop. So our work is based on one of the most important work here is from the Allen Institute. In their work, so they are looking at the primary visual cortex neurons. The mouse is sitting on a wheel that is doing an image change task. So the mouse is presented with a string of images with a fixed interval. At some point, those images are gonna go omitted. So there should be an image here, but it's omitted. What they found in this work is that it's actually and the VIP and the SST neurons are mostly stimulus driven. So they just activate by the stimulus. But for VIP neurons, there is this ramping dynamic during image emission. So there's an image here in the yellow box, and there should be an image here at 0, but it's gone. Then VIP neurons gonna keep ramping up until the next one. So this result is interesting. So we want to know what exactly it is. Basically, people say that this is a prediction response because these dynamics look like they are reset by the stimulus, and then they are expecting the next one until it comes. So if the next one comes, it gets reset again. So but another alternative hypothesis that they just encode the time information. So if the interval get longer, they ram further. If it's not, it's just a little bit. So these 2 hypotheses are confounded in previous studies. We want to know what exactly it is. This is a question we want to address in our paper. So whether these neural RAMs encode temporal signal of the encode elapsed time, previous elapsed time, or it was prediction related response. Is everything clear here? Okay. Then let's set up our question further. And so given a stream of which sensory stimulus, each black box here it represents one stimulus presentation. And we have an interval between each sensory inputs. So at the time t here, the neural activity, whatever it is, it can represent two things. One is just the elapsed time. So this is what we call temporal signaling, which means they are looking backward on how much time it already passed. And this is one hypothesis. They just encode time information. The other hypothesis is the temporal predictive processing, which means they are expect the next one. They are looking forward. So under this hypothesis, there are one important thing here is the temporal prediction error, which means if a stimulus should happen here, but it's gone, or a stimulus should not happen here, but it's here. And these two kind of unexpected stimulus gonna introduce temporal prediction error. They're gonna update the internal model on when the next stimulus is gonna come. This is what we call temporal predictive processing. So in summary, these two hypotheses means one, temporal signaling means the neuron is gonna looking backward. Temporal predictive processing means the neuron looking forward that expect the next one. This is what we want to address in our paper to distinguish these two hypotheses, whether the neuron encode time in temporal signaling or where the temporal predictive processing. Is it clear for everyone here? Okay. And to address this question, this is our setup. So a mouse is happy and sitting on a rig, and we use 2-photon calcium imaging to study their neural activity. And there will be a monitor in front of the mouse that presents a visual stimulus, visual and auditory stimulus. So this is our actual setup look like. And there is just a stimulus from the monitor and the speaker. And then there will be an interval between each stimulus presentations. We are looking at parietal cortex and the visual cortex. The parietal cortex, the visual cortex, we are actually looking at the PM area, so it's actually another higher visual area. And we are using transgenic mouse, VIPT tomato mouse, that we inject CNS precam. So under the microscope, those red neurons are the VIP neurons with the TT tomato labeling. And the green neurons are the non-VIP neurons that express GCAM. So we are looking at their fluorescent signal to study their neural activity. This is where we study VIP neurons and the excitatory neurons. And another mouse line, we use SST mouse, SST mouse. So we expect GCAM only in the SST neurons. And we're looking at these two different areas. These are some example Poisson traces for the three different neural cell types. Then the key part of our experience is to design this interval. Here, whenever we say trial, we define it as one stimulus presentation. Basically, it's the auditory and the synchronized auditory and visual grading images from the monitor and speaker, and following by interstimulus interval in between two stimulus. And this interval matters because it's the key part of the temporal processing. Then we designed these three different paradigms. The first one is the short-long sessions. when we present the stimulus with a fixed interval 1 second, and we repeat it as a block, then we switch to long interval block with 2 seconds. The key idea is that within each interval block, when we repeatedly present this interval, the mouse may form a prediction about the interval. And when we switch this block, they will update their prediction. So there will be a prediction within a block, and there should be prediction error across block switch. And this is just the hypothesis. Then we have what we call the bit jitter sessions. And this is to study if there is any prediction or prediction error, how it's related to the environment uncertainty. So during this block, the interval between the stimulus is 1500 milliseconds. And during jitter block is completely randomized into 500 to 2500 milliseconds. The key part here is that we have what we call the deviant intervals that is lying in the black dots here. They are 500 milliseconds or 500 milliseconds. So in the bit block, these deviant intervals are completely outliers, so they are very easy to notice if something gets unexpectedly longer or shorter. However, in the jitter block, because it's within this range, is expected. So if there is prediction error for these deviant intervals, they should be much larger in the bit block than the jitter block. So that's the idea. So, this session is to study whether there is really prediction or prediction error or not. Then we have the random interval sessions, which we just present images with a whole bunch of random interval between this range. They are uniformly distributed into 500 and 2500 milliseconds. And the idea is that if it's not prediction or prediction error, but just a temporal signaling, we should still see when the interval is completely randomized because there's nothing to predict here. Yeah, is everything clear for everyone? Okay. Then before we look into our real result, I want to show you how our method works, especially the clustering and the temporal resistive field models, because that's the key part of our paper. So if you look at how the neuron gonna respond to the stimulus and the interval in between them, it's gonna look like this schematic. So as you may see here, one part of the one category of neurons gonna get inhibited by the stimulus and then start ramping up. So this is more like the VIP neuron we have seen before in the other paper. And then this part of neurons, they are activated by the stimulus and then ran down. And they are more like the excitatory and excitatory neurons that get activated by the stimulus. But they are also having different dynamics. For example, this is dropping faster than this part. So when we're looking into our data, we want to categorize this different type of ramping activity and want to capture their ramping kinetics. This is why we use our clustering based on the GLM and the temporal resistive field model. So given a set of neurons, each row here is one neuron respond to the gray blocks as the stimulus. Then for each neuron, we will run a GLM model. It is very simple GLM. It's just nothing new at all. It's just a normal GLM. So the GLM gonna give you the kernel of how one neuron responds to one stimulus, around a stimulus. if you look into their GLM kernel, it's gonna look like this. So for example, there's a stimulus here. This neuron get excited by the stimulus and then going down. This neuron, on the other hand, they ran up between a stimulus and then ran down when after a stimulus. So on and so forth for each neuron, we feed a GLM to all of them to each run. A summary of how this neuron respond to one stimulus and the interval in between them. So to capture their ramping activities, we feed a temporal view model to each one of them. This model is basically a convolution of exponential function and a Gaussian function, and the formula is going to look like this. So as you can see, as the result, there will be a ramping up dynamics and then going down after some point. And if you flip it, you can have another kind of response. So they're gonna run going down. And that's what we call run up and run down categories. So for each neuron, we fit this model to capture whether it's ramping up or ramping down after a stimulus. And within each subgroup, so for run up and run down groups, we're gonna capture how fast or how different in their ramping kinematics. So we run a clustering within each subgroup. And then in our paper, we always call these ran up neurons, and these are the ran clusters. So we're gonna look into how these clusters respond to a stimulus and an interval. So far, is everything clear here?
Yes.
Sure, thank you. Okay, and then let's look into our results. This is the result from our short-long sessions, and this is the long interval. So there is a stimulus here in the gray box. There's another stimulus here. Then this is the interval. Each row is one cluster. The first two rows are the ran up clusters. These neurons get inhibited by the stimulus and then start ramping up. These three rows are the random neurons, so they are activated by the stimulus and then ran down up during an interval. So when we say activate or inhibit, is related to a stimulus. When we say it's ran up or ran down, it's usually during an interval. And what we found is that our model can capture these two broad categories of neurons. And within each group, we can further distinguish them from their ramping kinematic. For example, this is going faster, this is going slower, even slower. So here, the takeaway is that we can just use this model to find different ramping dynamics. This is how it looked like during one interval. And I'm showing here the PPC result. So what we find here is that even we are studying the visual and parietal cortex, we found that the results are very similar, probably because they are both higher visual areas. And we don't see much different in the distribution of the cell type specific response, like the other. Because as you can see here, the VIP neuron and the excitatory neuron are mostly similar across all clusters, but SST neurons are mostly the stimulus activate clusters. And yeah, that's the main takeaway here. Then the next question is, we already see how it responds during one interval, during the long interval. So how it's gonna look like when we compare the response during a short and long interval. So when we compare their response, what we found here is that the response profile are very similar. For example, for these random up clusters, they are different in terms of their response amplitudes. So in the long interval, as shown in green, is larger, but in short, it's just getting is smaller. And this is the same for all of the clusters. So what we found here is that the response does not differ within each one, but their response amplitude gonna change according to the interval that you see. Then this is how they're gonna look like to one stimulus. Now the question is there any prediction within a block or any prediction error could cause for a switch? The answer is we found that the neural activity gonna adapt or almost immediately with after a block switch. So let's look at this row, for example. Before the for the blue line, they are the response during the short interval block. Then there should be stimulus in the yellow line, but it's omitted. So in this at this point, it changed to long block. We can see that this adaptation is very fast, almost in one trial, and there's no adaptation like gradual adaptation within the block. And this is true for all of these clusters. For random clusters, they are like almost instant. And it is the same for the random cluster as well. And here is the same when we switch from long to short block. Our quantification further confirm this, because you may say that there may be some adaptation after when we expose the neurons to the stimulus many times. So for example, in this cluster, in the late epoch of the block, the response may go lower compared to when the response early in the block. But actually, this is not very different. And then we found that this, again, the transition is almost instant. So the conclusion here is that there is not much prediction and prediction error during our experience. Is everything clear? Okay. Now, based on the previous short long sessions, as we don't see much prediction of precision error, this street system gonna confirm again whether it is prediction or prediction error or not. Because the assumption in this session is that if there is any prediction or prediction error, it should be much larger in the fifth block than it's in the jitter block, because in jitter block is within the range and it is expected. But in the fifth block, it is completely outlier. Here is the result, again, from PPC neurons during long deviant interval. So there is, for fifth session, there should be a stimulus here. This is another stimulus. This interval is 1500 milliseconds for fifth block. But for jitter block, it could be anywhere. So this is the white box, meaning that it only presents in the fifth block, but not the jitter block. Then after this stimulus, there should be another stimulus here at 1500 milliseconds after the last one, but it's gone. Instead, it goes to 2500 milliseconds, which what we mean late stimulus presentation. Then we compare the response during fifth and jitter block. Fifth is in purple and jitter is in blue. What we found here is that the response during this deviant interval are completely overlapped for all of the cluster. So whatever they are ramping up or ramping down, their response are not different at all. If you look into whether you can decode the distribution, because you may say that they are just the same when in terms of their mean response, but there should be some difference in their the variability. But if you run a decoder during this interval to see whether you have any difference or not, what we found here is that the decoding accuracy is almost the same as the chance level. That tells you that there is no difference in their response comparing bits entry the block. Then we look into their population dynamics from PCA analysis. For the whole population of neurons, we run PCA and project them into three-dimensional space. And what we found here is that their population dynamics are very similar. So the dynamics should start here. There is a stimulus here in the gray box and then goes around. The stimulus should happen in the yellow point, but it's gone. Instead, it happened in the red dot, and it goes around. This is the case for the bit block. But for jitter block, it's a very similar thing. It goes around, and that. So even in the population level, there's not much difference. So what we conclude here is that their response are completely identical across this entry, which means their response is independent of the uncertainty of the interval distribution. So there is no prediction and prediction error, but instead they are just a temporal signaling, which means it confirms our another hypothesis. This result is the same for short deviant, so there should not be a stimulus here, but it present earlier than expected. When we compare their response, completely is the same as before. They are mostly overlap. Then when we run the decoder just like before, again, the decoding accuracy is just around the chance level. And the population dynamics does not look different at all. And this session paradigm just confirmed that there is no prediction or prediction error. It is just temporal signaling, and the neuron just encoded in that time. Is everything clear?
Yes.
Then you may say that we don't see any prediction error because the mouse has seen this session many times. We run this kind of session like 10 or 15, 20 days. If they receive 1 hour session there, they already learn something about that. But because the session confirmed that there's no prediction and prediction error, then whatever is found in our bit splitter session should also appear in the naive mouse. That's when the mouse hasn't seen anything before, and there's nothing to predict. And that's why we have these random sessions. When we present the stimulus and the completely randomized interval in between them, there's nothing to predict. And this is presented to mouse, the naive mouse, just haven't seen anything before. What we found here is that this signaling still exists in naive mouse. In naive mouse. So this column shows you 4 different clusters. And these 2 are the random clusters. These 2 are the random clusters. These 2 kind of response already exist in their first state. So if you look into how they differ across states, they are very similar actually. From day 1 to day 5, they are mostly overlap. So if perhaps you may say that they learn this very fast, so they can develop this prediction within a few trials or very short period. So we look into their response during day 1, the very first day, when they exposed to these sessions. And we divide the session into the first 50 and the last 50 trials, and which is what we call first and last epoch. And we compare their response profile in these 2 periods. Then what we found is that they still exist even in the very first epoch, they still exist. And these clusters are very stable across days. So it's not like they are the fraction of those clusters go from 0 and go up to some big value or something. But they just remain pretty stable and perhaps some change across days, but they still already exist. So the main takeaway here is that these neural ramps already exist in the very first day of the session. So this kind of temporal signaling is intrinsic and they do not need to be learned. So there's nothing to do with the prediction or prediction error. And what we found, same thing in the random sessions. What we found here is that they are just following by the relaxation process framework. So whenever this is the same for clusters as before, there is a stimulus here. Then we bin the stimulus into 4 different bins. So there is a stimulus here, here, or here. So for each cluster, let's look into this cluster first. They get inhibited by the stimulus and they start to ramp up. If the stimulus present here, they will get inhibited again and then ramp up. If there's not, they just keep going the same, keep going up and following the same path until the next one comes and they get reset. If you look into the population dynamics, there is a stimulus here, then the whole population is gonna evolve following the exact same path, exactly same trajectories. If there's stimulus presented earlier, they get resetted. If there's not, then they just keep going until the next one comes. And whenever the stimulus comes, they're gonna get reset by the stimulus. So again, in this random session, what we found here is that they just the neurons just encode time information. It confirmed our hypothesis of a temporal signaling instead of the predictive processing framework. Is everyone okay there? Okay. Then if we confirm that they are following the temporal signaling framework, they should encode elapsed time information. And this is how we confirm this. In our short long session, and this is the log during a long interval. Again, the same two big group, the two major category of neurons. The random neurons are here. The random neurons are here. Then we run the time decoding analysis, which means we are trying to decode pairwise time during an interval. So at 0, there should be a stimulus here. If at 2000, there should be another stimulus here. We are trying to decode time information during this interval. What we found here is that you can actually decode the time information very well for some cluster, for example, for these two. And for some clusters, for example, these two random up clusters, not so much. And if you fit the temporal receptive field model for them, this is how they're going to look like. So there's a stimulus here. This is the GLM kernel, and they ran up before a stimulus. These three clusters are activated by the stimulus and they ran up. We want to know what gives the difference in their decoding time their encoding time capability. So first we look into how the summary. This is basically the summary of this time decoding analysis. So again, they show you that these two clusters can decode time information in higher accuracy than the remaining three clusters. If we look into their temporal receptive field model, the parameters of the model, what we found here is that their decoding accuracy is consistent with their ramping dynamics. So for the green and blue clusters, they have different ramping speed and their temporal width. So our decoding analysis show you that while they encode time information during an interval, the parameters of their temporal perspective field gives the difference in their decoding capability. So here is what we have for today. So to summarize, and in this work, we study parietal and visual cortex in two-photon calcium imaging. We use three different experimental paradigms to distinguish whether the neuron is following temporal signaling framework or the predictive processing framework. So in our short long session, we found two main clusters, and then they have no adaptation within each block and immediate adaptation across both switch. There in our phase reader session, we found that their response during deviant interval is overlap for phase and jitter block. This kind of wrapping dynamics also exists in the very first day in the naive mouse in our random sessions. And this wrapping dynamics just following relaxation and reset framework. They encode time information, so we can decode time in high accuracy. And the reason of how they, the reason behind their decoding accuracy is their temporal perspective field models. So that's our conclusion. So the remaining questions in this work is that, so there is no prediction of prediction area in visual and parietal cortex in the higher visual area. So where the hell are they? Then we think that other brain areas such as like hippocampus or cerebellum may be involved in predictive processing, and that's our next step to study whether it exists in other brain region or not. So another point here is that the mouse is just sitting here doing nothing. So this is the completely passive behavior. If the mouse is just sitting here, doesn't need any behavior, then they really don't need to pay attention to the stimulus or the interval itself. It really doesn't matter. So we think that if there is active involvement in their behavior, so the mouse uses the interval to do something, there should be some prediction or prediction error signals, even in these two brain area. And this is what we are working on to change this into our behavior. And so that's it. Thanks for your attention.
Awesome. Thank you, Yicong. Do we have any questions?